Hello, everyone, and welcome to today's webinar, Understanding New Drug Applications, presented by Roe. I'm Joe Keenan, contributing editor to Fierce Markets, and I'll be moderating this webinar. Our speakers today are David Shoemaker, PhD and Senior Vice President of R&D at Roe, and Dr. Megan Spahala, Assistant Vice President of Operations at Roe. You can read their full bios on the right side of your window. Before we begin, just a few technical notes. This webcast is being streamed through your computer, so there is no dial-in number. For the best audio quality, please make sure your volume is up. You can find additional answers to some common technical issues located in the Help button at the bottom of your screen. This webinar is being recorded and will be available on demand within 24 hours after the event. We will follow the presentation with a Q&A session. Please submit your questions using the Q&A button on the left side of your screen, and you can do that throughout the presentation. All right, I think we're ready to begin. David, please go ahead.
Thank you very much. So by way of introduction, this is just the topics that we're going to cover today. I'm going to start with a pretty 30,000-foot view of an NDA, and then Megan is going to come in and give what I think is probably the most critical section of this presentation today, because it's about the integrated summaries, which I think receive too short a shrift in most presentations of this type. And then Megan will turn it back over to me, and I'll roll through the different documents in addition to the integrated summaries, the submission process and the review process, and finally, what happens at approval. So to start off with an overview, I think most of you are probably aware that the critical thing about your product is that it is safe and effective for the use in the population that is going to be receiving your medication. Benefit risk assessment is part of every application evaluation by FDA, and that is something that you can use beyond drug evaluation. You can actually use it in your day-to-day life as well. And finally, I'm also, in addition to the integrated summaries that Megan is going to talk about, I'm going to emphasize that the manufacturing process, while it's not forefront in everybody's mind, is often a stumbling block. And so I want to make sure that everyone's aware that that's a critical part of the process. So looking at what a marketing application is, it is FDA's license to you to sell and market your product. And it behooves you to give FDA an accurate representation of what your development program has shown. So the application has to be written very clearly, and it has to tell a story, both clinical and manufacturing and non-clinical, such that FDA can easily review and come to conclusions without asking a whole lot of questions. The fewer questions that you have to answer from FDA, the better, in my book. There are different types of marketing applications. We're going to be talking about specifically the NDA today. The abbreviated new drug application is also considered an NDA by generic manufacturers. And there are other biologic license applications, and there are obviously different applications of this type in other regions. But today we're going to focus on the U.S. NDA. And specifically, we're going to focus on primarily the 505(b)(1) application, which is an NDA for a new chemical entity or new molecular entity. And those are novel products that go through the development from discovery to marketing applications. There are also 505(b)(2)s, and as I said, ANDAs, which are also NDAs, but we're not going to be speaking about them today. So who's involved in an NDA? And I think this gets overlooked, is the totality of the development team. There are an incredible number of disciplines that must be engaged all throughout the process, and they generally have to participate in the marketing application as well. There's some aspect where you're going to need their contribution, and so if you don't have your team assembled right from the beginning, you're going to waste time identifying these components of your application preparation team. The format of the NDA, and you hear a lot of people refer to an NDA as a CTD, and it's not a CTD. CTD is merely a format that was developed by ICH in, I think, the year 2000 or thereabouts. It's been revised recently in a guidance in 2016, but it basically is a reorganization of the way an NDA was done back in the day, and it is harmonized between the U.S., Europe, and Japan, and other countries have adopted ICH as well. But it is a means for an individual company to submit a marketing application to several regions. Historically, that was thought to be done simultaneously in the different regions, but in actual fact, I mean, aside from the big pharma companies, a marketing application is rarely submitted simultaneously to both Europe and the U.S. It's done, months in sequence, oftentimes. The content of an application is obviously the data. That's what we're going to be talking about primarily today. The data is rolled up into summaries that make it easier for the FDA reviewer, and you also have to provide FDA with a history of your development process from the beginning so that they understand what agreements have been made prior to the submission of this NDA. And you all know that you're regulated by regulations as well as guidances and other regulatory publications. So the key decisions that a reviewer at FDA is going to make is, again, whether it's safe and effective for its proposed use in the defined population, whether benefits outweigh the risks, and whether the proposed labeling is appropriate. Manufacturing, again, enters in as an important part of this. Everyone thinks about clinical being the most important part of the NDA, and it does drive everything, but it is not. It is the data, ultimately, that is going to determine the approvability of your application. So one of the biggest mistakes that we see, having done, several dozen of these personally, and we do 2 or 3 of these a year, are the planning effort that's put into these applications. Typically, you should start planning your NDA at proof of concept, and most companies are loath to invest that much time, but it is driven obviously by your clinical proof of concept. But there are obviously supportive non-clinical studies that must be completed, CMC and quality, or CMC or quality issues that need to be conducted and evaluated, and regulatory strategy that all need to be folded into a target product profile so that you can determine the roadmap for your development. Waiting longer than the proof of concept of your therapeutic is definitely possible. You just lose efficiency. Also, as you start planning your NDA, it's good to start documenting the decisions that you make so you don't revisit these decisions continually. Having a decisions log does not require you to follow the only decision that you've made, but it creates somewhat of a higher energy of activation for you to revisit that decision. Again, safety database needs to be constructed. Data standards is something that we spend a lot of time on in our shop, because most companies don't give this the effort that is needed to allow a smooth transition to an NDA preparation. And then obviously you're going to have regional requirements that Megan will even talk to in her section of the talk today. So success factors are a coherent registration strategy, having no gaps when you ultimately submit your application. Your document should be well written, and it should be QC'd and QA'd so that there aren't obvious errors that the FDA picks up on and begins to dig deeper because they find inconsistencies. And this is all made much easier by a competent program manager who keeps everyone on the team operating at maximum efficiency to their defined timelines, so that you're going to have bumps in the road regardless. But the more transparent you are with the entire team on when everything is coming together, the more efficient your process will be and the more likely you are to meet your timeline. Again, this is the organization or the format that I talked about that was promulgated in 2000, the CTD. Module 1 is different for Europe and Japan and the US. Module 2 through 5 are identical in the 3 regions, with the exception of some special documents like the ISS and ISE that Megan is going to talk about today in the US. The label, whether it's a target product profile or it's an annotated package insert or it's the final package insert, is the absolute distillation of your entire NDA. The target product profile can be used very effectively in development. The annotated package insert, you are required to annotate or footnote every statement that you make in your final label. And so it's good to be thinking about what data you have that supports statements that you want to make in your label. I am forever talking with companies that want to say something in a label when they have no data to support that statement, and it just can't be done. FDA won't let you do it. And the package insert or prescribing information is the ultimate goal. And this is just an example of a table that we use in our NDAs, and you can see that this is the indications and usage section of the label. The product is indicated, and you define what the indication is in the draft text. And then over here on the right-hand side, the annotation module or reference listed drug PI, if you're doing a 505(b)(2), you have to reference where the facts are that justify your statement. Absence facts or any justification, you are not allowed to make a statement. And that's an important issue that's lost on many people. So this is the organization of the CTD. Again, Module 1 contains the label, but aside from that, it's regional, depending on what ICH region you're submitting your application to. I will go through later the U.S. components in Module 1. And if you look at the CTD and MAS, the data resides in Modules 3, 4, and 5 for CMC, non-clinical, and clinical data. And that's where the original reports, the CSRs for clinical, the stability reports for quality, and the toxicology reports for non-clinical exist. Modules 2.6 and 2.7 are distillations, but it's still largely data. So that's where the data resides. And then FDA allows you the ability to create opinions surrounding this data in the summary. It's not, ad libitum your interpretation, but you are allowed some leeway, whereas in the data sections of the CTD, it's just the facts, ma'am. This is your interpretation. It's where you can begin to craft your story. So if Module 1 is regional and it differs between Japan, US, and Europe, Modules 2 through 5 are the CTD proper. And again, if you write this once, you can then use this section of the CTD in multiple regions around the world. So if you look at the Module 1, again, the label again is the distillation of all of this that's working up to the summary in Module 1. So now I'm going to turn it over to Megan, and she is going to talk about what I think is too often overlooked and not done soon enough by many companies preparing their NDA.
Thanks, David. So just to recap, the NDA includes all information about your product from its inception through clinical testing. And the integrated summaries of safety and integrated summaries of efficacy are limited to just the clinical portion of your product development. But they tell the story of the data across your whole clinical program. So similar to how your clinical project needs a statistical analysis plan, your ISS and ISE also have statistical analysis plans that are needed to describe the integrations and analyses that you're going to perform. Now, these statistical analysis plans are not just a regurgitation of what happened at the study level, because it's a separate analysis with a separate purpose. And we're going to talk about the ISS, ISE, and the purpose and goals for the next several minutes. So your ISS, as you know, your phase 2 and phase 3 studies, they are designed to establish efficacy or effectiveness for your product. Most aren't designed to test safety or to identify adverse events that are predefined with a level of specificity. So really what your goal of your ISS is to combine the safety data of your program to describe the safety profile of your product. This can include analyses of safety-related events, estimates of risk over time. You'll need to look at different possible subgroup differences in your safety, and also it will help you identify risk factors associated with the events and enumerate them to the FDA. So what we want to make sure is there are things that we expect a reviewer will look at with regards to your integrated summaries of safety. And in 2005, the FDA put out their safety review guidance. In 2010, CDER additionally put out a good review practice, which provides a lot of insights into how the agency will review NDAs. At a minimum, you will expect that your reviewer will look at serious adverse events that could be related to the drug, common AEs that could be related to the drug, and then factors that would predict the occurrence of these adverse events, as well as exposure. So exposure looks at the duration, the amount of time, the population, and how long you followed your subjects up in your clinical studies. Before we get into the meat of what you should be looking at and questions you should be asking about in your ISS, there are key considerations and 3 guiding principles that you should always keep in mind related to an analysis plan you're creating for your integrated summaries of safety. First, you want to produce reliable estimates of safety that describe your whole profile. To do that, you we'll talk a little bit more about how to define your parameters of interest, but once you have those, you also want to include AEs of special interest and different populations of special interest. A lot of these are going to be dependent on your indication or on the class of your drug product. Additionally, you have to, all of the ISS is basically exploratory in nature when it comes down to the statistics that you use. There's very little formal statistical testing, i.e. testing in an adequate, well-controlled environment where the p-value has anything other than a descriptive meaning. But you still need to look through all of these. You need to test different things, because what you want to do is show the agency that you have identified trends and issues and that will help with your risk-benefit statements later on. So when you start looking at your ISS, as David said, this really takes a village. There are a lot of people involved in making sure that the messaging that you have in your NDA are appropriate for the indication that you're trying to get approval for. So there are 3 key questions that can really help your whole team move forward in what's best for the ISS. So this is when you get your clinicians, your statisticians, your medical writers all in a group, all in a meeting to talk about these 3 key questions, and these questions should be asked in the order that is on your screen. So first is, what are the safety parameters of interest? You'll have to look at the key safety messages you'll want to go in your label and define what parameters could then help you with that messaging. Once you know the parameters, you want to ask how to present those key safety messages to define the overall safety profile. And then once you have that, you can talk about how to best characterize the information from the studies in the program. That was really quick, so let's get into each question individually to have a better idea of what your team should be discussing. So there's a lot specified in agency guidance on what safety parameters you should look at. There are also going to be AEs of special interest based on your indication or based on the class of drug or your compound. And there are also going to be things that come up throughout the studies that allow you to want to look further into it to make sure you're not seeing trends across studies. There are also, as you finalize your program and you're getting toward the end of your last phase 3, you're going to be reviewing the data to identify any safety parameters that have come up that were unexpected at the start of the program. So when you look at the different parameters, typically you'll see things about exposure, and the agency is going to expect exposure data, data on concomitant medications, deaths and other adverse experiences, laboratory measures and vital signs, and then other investigations that are either specified in an agency guidance on your therapeutic area or otherwise, or have been discussed through either your pre-NDA meetings, your end of phase 2 meetings. You're going to want to look through all of that to make sure that you're investigating appropriately. Although all of these are quite common, the level of emphasis for each is really going to be very dependent on your specific compound and the class of drug and the type of indication you're going for. I can't stress enough how important it is for clinical and stats to come together to address if these parameters are going to be the most effective to develop your key messages. Once you've prioritized the measures and you have them in place, we can start moving to question 2. So really, this question is, once you know what you're going to look at, you need to define how to present the key safety messages and overall profile. So when you're looking at this, you need to think about what should the analysis and presentation approach accomplish. And you're gearing all of this to how the reviewer is going to see this data. So you want the reviewer to get a true value of the parameter of interest. And you also want to make sure that there are meaningful comparisons. This could either be between active groups and placebos, or it could be between any comparator group that you've had in your phase 2 and phase 3 trials. So how do we do this? So how can we, with the parameters, how do we make sure that we're characterizing them appropriately for the agency? There are a lot of different choices here. And the methods listed to the right are common in epidemiologic analyses, and they're also very common throughout an ISS. But what you choose is dependent on the parameter of interest. So you might want to use a crude rate or just a proportion. So if you want to know that 35% of the subjects on this across the program had a certain adverse event, that might be worthwhile, and then you could just use a proportion. You might want to use an incidence rate. If time is a factor, for example, if you have a vaccine trial, you're going to get erythema. But what you wouldn't want is erythema after 2 weeks after the vaccine. So you might want to look at certain time-delimited functions, and then you would use an incidence rate. Or there might be an instance where you wanted to use, where a subject could get multiple events throughout time. And if you want to take into account not only the number of subjects who had the event, but the number of events that happened, you'd want to look at a total incidence rate. So once again, as you're going through your parameters, what you truly want to look at is the message you're trying to state, what you want to look at, and then your statistician can help you with what type of measure you'll look at to make the most impact in your messaging. Once you know how you're going to characterize the safety parameters, you can also look at how you're going to compare the two treatment groups. Again, one has many choices. Your statistician is going to be able to tell you what is best for your messaging based on what you've chosen as your characterization. And I think that here you have to remember that these comparisons differ from formal comparisons that would be in any efficacy setting. So they are made for the purpose of assuring that no safety signal is missed. They're not necessarily made to show that there is a treatment difference or a treatment effect. You're wanting to give confidence that you've looked at your safety parameters and that you're not missing anything as far as a trend or a safety issue. So with these key parameters that you want to look at, you also need to ask about questions of special interest. We've come up with methods to characterize our parameters. We're going to compare them across groups. But based on your indication, there could be questions that you want to consider. And that could be, are there specific adverse experiences that are of clinical importance? Are there known adverse experiences with your drug or with drugs in your class that you should be highlighting for your risk-benefit statement? And also, you don't want to give the agency everything. You have to focus on what is important for your compound, for your indication, for your therapeutic area. One of my colleagues routinely says, don't send a mine and tell the agency to find the diamonds. You need to focus, and you need to tell the agency about the diamonds. So there are going to be good things and there are going to be bad things, potentially bad things that come out of your integrated summary of safety. The biggest thing is that you show the agency what you have and what you found, be transparent, and then any of the things that could be deemed questionable can be addressed in your risk-benefit statements. You also, when looking at the parameters to use and how to characterize them, you need to consider some special challenges that can pop up across your program. So these are things like the variable lengths of follow-up and exposure across the different drug trials. There might be some studies that are uncontrolled. There might be, while the others are controlled and have a placebo arm. You could also have an unblinded study. All of these are going to add in different levels of variability to your point estimates, which then just need to be able to be described and thought through. So the easiest, or the earlier you discuss these special issues that can pop up, the better you'll be and the better messaging and narrative you'll have in showing your data to the agency. So now we have the parameters of interest. We know how we're going to characterize them. We know how we're going to show differences across treatment arms. And now we have to go and paint the picture of our safety profile. And the biggest thing is that we want this to be an objective look at the safety profile. As David said, there are other modules of the NDA that can get into more building the story. This is what the data is showing and only what the data is showing. So you're under a regulatory obligation to present all of the safety data. This could include all studies in the database, but you're under no regulatory obligation to pool all of the studies for all of the analyses. That's really ultimately a sponsor decision. However, you have to keep in mind that the FDA and any advisory committee panels that you might go be part of are convinced of the validity of the strategy. So really what this comes down to is how do you pool your data? You would pool your data because, once again, your individual trials are not really set up as far as the sample size is concerned to show safety trends, especially for more rare incidents, rare events. And so the more data you have, the more precise and reliable your estimates are going to be. That will allow you to better describe your overall safety profile. Also, as you get an increased sample size, you're allowed to explore where there are possible interactions. This could be a drug demography interaction, a drug disease interaction, a drug-drug interaction. Having a larger sample size can allow you to look into these subgroup analyses. So you want to combine your data in the most valid way, and your safety message should drive the pooling strategy. So you don't want to pool just for pooling's sake. You want to make sure that you're taking the message into account. You also want to make sure that you're pooling with an effect to have transparent results. So this isn't the time if one of your pivotal trials showed that there was a safety signal. What the agency isn't expecting is for you to pool everything to mask that one study's output. You want to be transparent in what you've seen, and in that particular case, the agency will likely look at an individual study level anyway and see that the one trial had a different safety event status than the others. So what are some factors to consider about pooling? The biggest thing is that there is variability across your studies. And so the more differences you see across your studies, the more variation in your parameters it's going to be imposed on. So you have to look at the design similarity. Are your designs similar? Did they have similar visits? Did they have similar visit windows? Were the doses similar? Did you follow the subjects up for the same duration? Did you dose the subjects for the same duration? And then the type of control you use, whether it's placebo-controlled, active-controlled, or an uncontrolled trial, the geographic region in which your studies are conducted, and then your populations all need to be taken into account when you're developing your pooling strategy. You want to make sure that it makes sense to show what is going on as far as the safety profile, but also makes sense in regards to the population that you're trying to get on your label. So to recap, on the ISS, we've provided some information that is useful in what you need to think of when creating your integrated summary of safety. And we've talked about 3 questions that you should work through that can help provide a roadmap to creating a complete safety analysis of the product. The ISE is, you can ask very similar questions to the ISE as the ISS, and I would actually ask that you do, because once again, you want to focus on your parameters of interest, how you're going to describe them and characterize them, how you're going to show differences, and then how you're going to pool. But the ISE is really focused on integrating summary of data demonstrating effectiveness rather than safety. And you want to focus on your claimed indication. So you want the data to support your dosage and administration. You also want to look at subgroups to make sure that there is efficacy across different subgroups, or if there's not, you can discuss that further in as it might be a difference in your label claim. So your primary objective for your ISE is to get precise estimates of your primary endpoint, which is very similar to the safety. If you are able to pool some of that data and pool some of the information, your estimates of effectiveness will be more precise. You also want to look at key secondary endpoints, assuming they support evidence for your primary. And then the agency is expecting to see subgroup investigations to examine the consistency of the effect. So here you're going to rely on your adequate and well-controlled trial. These are typically your phase 3 studies. However, they don't necessarily need to be your phase 3 trials depending on your program. Phase 2 trials that are not adequate and well-controlled typically don't control for your type 1 error rate. You tend to have a lot of multiplicity. You're having multiple comparisons throughout your trial, and so these weren't set up to be adequate and well-controlled studies. But throughout your ISE, you want to focus on your primary endpoint or co-primary if that's how your program was set up. And as I said, you want to look at 1 or 2 secondary endpoints that have clear clinical relevance to show consistency of your product across multiple different endpoints. Subgroups of interest are important because the agency does want to see that consistency in effect. And once again, with a larger population, with the pooled data, you have more precise estimates of if there is any response based to any subgroups. The subgroups could be age, gender, or race, or something else that is important to your particular indication. And just like the ISS, everything is not okay. You don't want to throw in every endpoint, secondary and exploratory, that you've looked at across your program. Keep it simple, stick to the primary, and then 1 or 2 key secondaries. And then finally, the ISE is also where you're going to show your demographics and baseline characteristics to categorize the population, because you want this to be your evidence to support your proposed labeling population as well. And any differences that come out of the populations based on the program that you have should be addressed, and you need to talk about why you are or are not pooling your data. So I'm going to send it back to David to just give a quick overview of the rest of the NDA process.
Thanks, Megan. So hopefully by now you have an idea of the criticality of the ISS and the ISE to the success of your NDA. That is where FDA starts when they're determining the safety and effectiveness of your product. There also is Module 2.5.6, which is actually a high-level summary of benefit risks, which incorporates the meaningful elements of the ISS and the ISE into almost an abstract of why your product should be approved. So those are the seminal NDA documents in addition to the label, obviously. So now I'm going to briefly go through the other technical module documents and hopefully leave time for questions at the end. Just to reorient you again, there is CMC quality, non-clinical, and clinical cascading up to your label in this organization of the CTD. The clinical, Module 5, contains primarily of importance both the tabular listing of all clinical studies, and FDA has a very standard format for this table that allows them to look at your entire clinical program and see what was done in significant detail. And then your ISS and ISE will typically be in Module 5.3 along with your clinical study reports. If you have a very abbreviated development program in a rare disease, many times you can get permission from your reviewing division to put the ISS and the ISE into 2.73 and 2.74, but there are page limitations on those sections, which you will see. The ISS and ISE typically live in 5.3, and then obviously literature references. So Module 2.7, again now, we're moving up in the triangle into those documents that are allowing some interpretation on the part of the applicant. So you can begin to describe your data with what it means to you a little bit more than just giving blanket statements about what the data say. And the biopharmaceutics and analytical methods is primarily a CMC-driven document with elements about manufacturing and development of your product. Clinical pharmacology is really the clinical aspects of your product, how it's handled in the body, what are the pharmacokinetics, the effect on the body with what time frame. And then you have the 2.73 and 2.74 documents that I just referred to, that if they're not the ISE and ISS proper in a rare disease program, they are a distillation of the ISS and the ISE. And again, literature references, synopsis of individual studies. But this entire 2.7 module is recommended to be less than 400 pages in the ICH guidance. And so rarely does the ISS and the ISE make it into this because of the sheer volume of those documents. But I encourage you to talk with your regulatory project manager at FDA, because we just submitted Module 273 that is probably 400 pages alone. So they do bend the rules sometimes with different divisions, but you've got to get clearance. Following Module 2, non-clinical documents, you have the data and the interpretation in the Module 2.6, which is also contains a comprehensive tabulated summary of what studies were done, and then the messaging or the opinion resides in the overview, which is in Module 2.4. 2.6, again, tabular summaries dealing with pharmacology, and there are recommended formats for these tables, and ICH and the FDA has guidance on this as well, but typically broken down into pharmacology, pharmacokinetics, and toxicology. The pharmacology being those experiments that demonstrate your rationale for development in the first place, and the pharmacokinetics being how your drug is handled in animals. And toxicology, I think people are aware of. Now, this is the organization of Module 2.4. Again, it's an overview, so it allows some interpretation for these different disciplines, whether it be pharmacology, toxicology, or clinical pharmacokinetics in animals. This is all animal studies that are being described here. And the mechanism of action, it's not a absolute that you must have a mechanism of action for your product, but FDA wants to make sure that you at least did the due diligence necessary to examine what the mechanism of action is for your product. And it also includes primary pharmacology, secondary pharmacology, and safety pharmacology. So before you ever get to the toxicology studies in your program, you should examine some of the safety parameters in the animals that you're testing in your pharmacology studies. And then the pharmacokinetics, how the drug is metabolized, and ultimately the toxicology. The quality module 3 is pretty straightforward but pretty detailed. So they break it down traditionally into the drug substance, which is your active pharmaceutical ingredient, and the drug product, which is the final material which is injected into humans. This is just an idea of the outline of the table of contents for the drug substance. None of these documents are that long, but they're extremely technical and need to be reviewed and presented accurately to the FDA reviewer. This is a very, I'll say, attention to detail part of the, it's all attention to detail in the NDA, but chemists tend to be over the top, so these documents have to be pristine. Similarly with the drug product, the final material that is injected or swallowed by the humans is described in Module 3.2p. Best practices for preparation of the CMC or quality documents. This is always occurring at the last minute, it seems. Clinical is done and wrapped up, and there are still CMC activities that are ongoing. So whether it's a last lot of material or it's some contaminant that has suddenly sprung up, be prepared to make last-minute changes in your CMC section. The document 2.3 is a summary of Module 3, and this has to be written after all the work is done and all the data is prepared in order for it to be, up to date. This is the organization of Module 2.3, the overall summary. Again, just breaking down those complex tables of contents that I showed you earlier into a brief narrative. Again, Module 2.2 and 2.1. 2.1 is simply the table of contents, and the CTD introduction is an abstract for the entire NDA. The Module 2.5.6 is an abstract of the benefit risk. This is an abstract of everything that's in your NDA. So it should be written last. It should touch on all the critical messages or the label components that you want to point out to the FDA. And this can be a leading document, but if it gets too biased, the FDA is going to stop reading. So it needs to be a well-written document with some slant, but not too forceful. And then Module 1 is a lot of information for the region that you are submitting your NDA. And these are just some of the documents that are included in Module 1. It's, as I say, not just forms, but it varies from the history of the regulatory correspondence and meeting information that you've exchanged with FDA, the patent information, which is obviously important for generic manufacturers who are looking to follow your product into the market, and you need to apply for your proposed proprietary name well in advance of your submission date. These are box-checking exercises, but they're not without a significant amount of work, especially things like if you're developing a opioid with a anti-abuse component to it. There's an entire set of studies that have to be done and included in the NDA. A medication guide, if your product is not inherently clear to the patient, you need to give the patient some help. The user fee waiver or the user fee has to go in before the NDA. If you're not going to pay that fee, you have to justify. Letters of authorization from people whose data you are accessing. And if you do have a life-threatening disease, you can include justification for a priority review so that your application is reviewed in 8 rather than 12 months. So that includes the documents. I'll now quickly go through the submission. This is just a routine electronic publishing and submission process. It starts out with report level publishing. It goes to submission level publishing. That has to be approved by the sponsor of the drug who is going to submit it to FDA, and then it's submitted to FDA via the electronic submissions gateway, and a receipt and acknowledgement will be obtained within hours after submitting your NDA. To give you an idea of what these NDAs comprise, this is the total files, ultimately down to, this is just a recent example that we have. The volumes are fairly significant if we were still doing it in paper. And the hyperlinks is a job in and of itself, making sure that all those hyperlinks go to the right places. The fiscal year 2018 user fee is on the order of $2.5 million if it's an original NDA. If it's a 505(b)(2) and you don't require clinical data, you can get away for a simple $1.2 million. The review process has changed in the last 5 years, and FDA on its website has a very good diagram that demonstrates when the submission is given to FDA. If it's a priority review, this whole process takes 4 months fewer than a standard review. But that has to be for a life-threatening indication that you're typically going after. Otherwise, you'll submit your NDA, and typically within the first 2 months, you will hear back from FDA that your application has been filed and is in review. And the review takes, obviously, several months. There is often a mid-cycle review meeting. It's not mandatory, but it happens on, I would say on average 75% of the applications where FDA will ask you to come in to resolve some issues that they've turned up during the review. And then finally, if it is 8 months, priority review will be finished and a review decision will be issued. Otherwise, you will most likely undergo a advisory committee meeting and have another meeting with the agency. The wrap-up meeting is one that allows you to begin talking about your final label, and official action will be taken prior to your PDUFA date, which will be for a standard review at the end of month 12. And there's a lot of activity here. FDA, their team works on one NDA at a time, it seems. So as soon as they get you information, it behooves you to turn that information around by the deadline that they give you, which is typically 24 or 48 hours. The approval process is fairly straightforward. At the end of those 12 months, you'll be told that you are approved, and that is really the goal of your NDA. And FDA is doing a much better job at approving applications during the first cycle rather than issuing a complete response letter. But if your NDA is deficient in any way, shape, or form, you will receive a complete response letter with a list of deficiencies that you will be required to answer and resubmit that as a complete response to FDA at whatever date you finish all the work that they've given you to do. So a first cycle approval is what is the goal when you're submitting an NDA. And with that, I will turn it back to Joe and ask him to moderate the Q&A session.
Okay, David, thanks. We'll go ahead and move on to the Q&A. I think we have enough time for a couple questions. So, David, Megan, I'm just going to kind of throw them out there. First question is, what percentage of NDAs are approved following the initial cycle of review?
Yeah, that's what I made reference to. I think it's up to around 75% now. 5 years ago it was down in the 50% range. So it was, I think FDA is taking it seriously. They're trying to give more information to the development teams earlier on, meeting with them more frequently. Certainly breakthrough designations have assisted in upping this with the communication on and sometimes an almost monthly basis. So, but right now I would say that it's up around 75%.
Can you give a brief outline on the major differences, if any, between a BLA and an NDA?
Yeah, I think the differences really are not in the review period. Obviously, CBER and CDER are not identical centers, so they have their own inclinations. I think the CMC section is typically much more complicated on a BLA, where you have a biologic that you're examining, and you just have a lot more biology to deal with. Whereas on the CDER side, with the drugs, it's typically a small molecule interacting with a receptor, and the downstream biologic effects are generally can be less complicated than with a biologic. There's also the immunology associated oftentimes with most biologics. So I would say the CMC is probably the biggest difference. That being said, BLA sometimes can be very quick if it's a very specific monoclonal antibody targeting a very specific receptor. So it's not a black and white, but frankly, I would bet on a NDA review being quicker.
All right, this looks like this might be our last question, and it's a twofold question. Does Roe have any experience helping companies prepare NDAs for live biotherapeutic products? And if so, can you comment on any ways the NDA preparation process for LBPs is distinct from the process for small molecule drugs?
Yeah, I think it's infinitely different. We do not have any experience with live biologic products. We tend to work with dead biologic products if they're alive at all. I mean, we do work with gene transfer and gene therapy products, which in some sense are alive in as much as they can be integrated throughout the body, and that's a safety concern of FDA. But live viruses or things of that nature, we do some, I would say we've done a fair amount of vaccine work, but I don't know if that's what you're specifically talking about in terms of a live biological product. But vaccines
David, I'm
are
going to try and squeeze.
Okay.
If you wanted to continue on, I was just going to squeeze one more question in if we could.
Sure.
What is the approximate cost of each FDA hearing?
Free.
Well, that made it easy.
There's no charge. It's just getting a face-to-face meeting with FDA. If you don't have the justification to meet with them face-to-face, they will many times relegate you to a teleconference, which in some cases is not bad because they're able to put you on mute and discuss things and change their decisions, whereas in a face-to-face meeting, they are extremely reluctant to change their minds on any thing that they decided before they walked into the room.
All right, that finishes our Q&A session. I'd like to thank everyone for attending this Fierce Markets webinar and submitting so many great questions. I'd also like to thank our speakers for participating and Roe for presenting today's webinar. As a reminder to the audience, the webinar has been recorded and you will be able to access the recording within 24 hours using the same audience link that was sent to you earlier. Thank you again for joining, and we look forward to seeing you at future events.